title of my talk is a new searching algorithm for identification and quantification of environmental microplastics by Pyrolysis-GCMS. The background of this research is that microplastics of less than 5 mm in size in the environment have various impacts on marine organisms and human health through the food chain. Conventional analytical methods such as micro-Raman and micro-FDR-ATR are suitable analytical techniques when the particles collected on the filter are visually identifiable. But they are not best suitable for mass-based quantitative analysis because they only measure the number of particles. On the other hand, Pyrolysis-GCMS complements the spectroscopic techniques as it can analyze polymer mixtures and does not depend on the particle size of the sample. However, due to the complexity of the acquired data, there is a need for a new searching software for qualitative and quantitative analysis of polymer mixtures or microplastics. Previously, we proposed the reactive pyrolysis, thermally assisted hydrolysis and methylation GCMS method. Its advantages are simplicity and high sensitivity detection of pyrolyzates in PET and polycarbonate sample. However, the disadvantage is that the reproducibility of some reaction products from polyurethane is poor and the error of quantitative analysis is high. To solve this challenge, we developed an analytical method to improve the reproducibility by converting diphenylmethane diisocyanate (MDI), a pyrolysate of polyurethane, into diamino diphenylmethane (MDA) using calcium carbonate as a catalyst. Then, using the analytical method, we developed a searching algorithm and searching software for the identification and quantification of environmental microplastics by Pyrolysis-GCMS. This is a figure of Pyrolysis-GCMS system. It consists of a single Q GCMS and pyrolyzer, packed GC glass insert, pre-column, separation column, and bent-free GCMS adapter. To backflash high-volume compounds derived from polyethylene, we used a multifunctional sampler that has a backflash function. A typical analytical procedure is to place a sample in a sample cup and drop the sample cup into the pyrolyzer furnace, and then perform flash pyrolysis in the furnace at 600 degrees C, then introduce pyrolyzates into GC column and run GCMS. The resulting pyrograms are analyzed by the software that we developed. In the previous study, we reported that in the development of reference polymer sample, the analysis reproducibility can be improved by dissolving solvent-soluble polymers in the solvent. Also, to reduce the weighting error of solvent-insoluble polyethylene and polypropylene, it is necessary to add and mix powder diluent to PE and PP samples. We found that the deactivated quartz powder was suitable for uniform mixing of polyethylene and polypropylene as a diluent, and the quartz powder did not affect pyrolysis of the 11 polymers alone. However, when 11 polymers were mixed and pyrolyzed together, only polyurethane did not produce its characteristic peak MDI. It is assumed that polyurethane reacted with the pyrolyzates of PET to form MDA and MAI, instead of MDI originally expected. It would be difficult to study using highly reactive MDI as the characteristic compound for polyurethane quantification, so we decided to allow MDA to be generated. By using calcium carbonate as a powder diluent, it was possible to catalytically convert MdI to MdA. Also, calcium carbonate was found to act as a catalyst in the pyrolysis of other polymers such as polycarbonate and PET. For polycarbonate, pyrolysis of bisphenol A, the main product, is promoted and the amount of IPP formation is increased. So for PET, benzoic acid is converted to benzophenone. Therefore, we use IPP and then BP as the characteristic compounds for the quantification of polycarbonate and PET. A reference polymer sample containing 12 different polymers were prepared as follows. The powder mixture was prepared by adding a large excess of calcium carbonate to the fine powders of polyethylene and polypropylene. The remaining 10 polymers were divided into 3 groups: THF-soluble polymers, dichloromethane-soluble polymers, and HFIP-soluble polymers, and polymer mixture solutions were prepared for each solvent. Using the prepared polymer solutions and mixed powders, the reference polymer sample was prepared in a sample cup for Pyrolysis-GCMS measurements. First, 4.0 mg of calcium carbonate, polyethylene and propylene powder mixture was added to the sample cup. Then 8 microliter of each of the 3 polymer solutions were added and allowed to dry. The content of each polymer in the sample cup is shown here. Since it was needed to prepare a calibration curve for quantitative analysis, we also prepared samples with 1/2 and 1/10 of this polymer content. When we analyzed the reference polymer sample, there were challenges with peak separation and shape. You can see many different pyrolyzed peaks on this pyrogram. Depending on the concentration and types of polymers, there is a possibility of secondary reaction among pyrolyzed peak, which leads to overlapping peak and abnormal peak shapes. To solve the challenges, we used a medium polar column and packed GC glass insert. The medium polar column improves peak separation. Without the medium polar column, the characteristic peak of nylon 66 is overlapped with pyrolyzed peak of polyethylene. Whereas with the pre-column, characteristic peak of the nylon 66, so cyclopentanone, is well separated from polyethylene pyrolyzed peak. Improvement of peak shape by packed GC glass insert. The top pyrogram is the one obtained using regular GC insert without packing material when pyrolyzing polyethylene sample. The bottom pyrogram is the one obtained using a packed GC glass insert. It can be observed that from C20 to C30 area, the peaks are broad in the stem and has lower peak height. You can see that the peak heights are improved when we use a packing material. Also, the peak shapes are sharper, as in the pyrogram here. So far, we optimized the analytical method and system. From here, I will talk about the data interpretation using the software that we developed. The analysis procedure is shown here. First, analyze each target polymer and store their characteristic pyrolyzates and ions in the library. And next, the characteristic pyrolyzates from the reference polymer samples containing 12 polymers based on the library are automatically identified. The data from several reference polymer samples with different concentrations are processed to create calibration curve. After obtaining data from actual samples, the software calculates the match quality and quantitative value. This slide explains the algorithm and how we created the library. In the case of polystyrene, since styrene dimer and trimer are characteristic pyrolyzates, their mass spectra and characteristic ions are stored. Then summated mass spectrum of these two pyrolyzates is stored as the mass spectrum representing polystyrene. The characteristic peak used for quantification is the styrene trimer for polystyrene. The other 11 polymers are stored in the same manner. Then the reference polymer sample, which is a mixture of 12 polymers of known weight, is analyzed. The software automatically detects the peaks of the pyrolyzates used to quantify each polymer and calculates the area and height. By measuring the reference polymer sample at varied concentrations, an absolute calibration curve is created. This is the basis for the calculation of the quantitative value. This slide explains how to calculate the match rate for each polymer for unknown samples. We use polystyrene as an example. Since the characteristic ions for styrene dimer and trimer are stored in the library, we obtain EIC from the unknown sample data. Peak detection is performed to determine the candidate peaks and obtain the mass spectrum based on the retention index and detention time. In this case, A and B are the candidates. The match rate is the similarity between the combined or summated mass spectrum of A and B and the summated mass spectrum stored in the library. The effectiveness of developed software algorithm was verified with two test mixture samples. One sample is a mixture of polystyrene, nylon 6, and polycarbonate. Another sample is a mixture of PVC, PMMA, polycarbonate, PET, and nylon 6,6. In the table, the dark blue color indicates the type of polymer that was mixed in the test mixture samples. As a result, we found that if the match rate is more than 80% and the quantitative value is about 10 times larger than the value of LOQ, then the polymer is most likely contained in the sample with a high probability. This is an example of the analysis of microplastics sample in the environment. We analyzed unknown marine plastic samples collected off the coast of Japan. The sample was provided from Professor Isobe in Kyushu University. This is a picture of the collected samples. The samples were ground to uniformity in a freeze grinder, and about 0.1 milligram of the sample was analyzed by Pyrolysis-GCMS together with 4 milligram of calcium carbonate. These are analytical conditions for pyrolysis and GC and NMS. This is a summary table of search results and comparison of the selected summated mass spectrum from the unknown sample and library database, and comparison of characteristic peak. For polyethylene and polypropylene, the mass spectrum is in a good agreement with the library, and the characteristic pyrolysates C21 and C9 are clearly observed, and quantification value is several times higher than the LOQ. So this indicates that these two polymers are present in the sample. For nylon 66 and PET, all the selected summated mass spectrum is in good agreement with high match rate. The quantitative value were very low, so although there is a possibility that they are contained in the sample, a more detailed analysis is required. For PMMA and nylon 6, the match rate was low. Look at the selected summated mass spectrum. And also pyrolysates were very observed, so it could be assumed that they are not contained in the unknown sample. This is a summary. We developed a searching algorithm and searching software for identification and quantification of environmental microplastics by PyGCMS. For qualitative analysis, the software uses characteristic pyrolysates and their selected ions, and provides the results in about 15-second processing time for data of 30 minutes GC runtime. For qualitative analysis, it can be done at the same time. In conclusion, the developed software was tested on different polymer samples and unknown environmental microplastic sample to validate its effectiveness. The last message is that when the environmental samples are pyrolyzed and the data are searched by the software, you need to keep in mind that search results are telling you the types of polymers. For instance, if the searching result tells polystyrene is contained, that means your sample contains polystyrenic samples. It may not be polystyrene only. Although the software assists the interpretation of your data, it will be important to keep that in mind to draw your conclusion. Thank you very much for your attention.
Thank you very much, Ichi-san, for the nice presentation. So we do have time for quick 2 questions. If there are any questions, I request the audience to post the questions in the chat box. And before I get any other question, I do have one question on your presentation. So you basically categorize the different polymers that are identified by the software using that probability percentage. So certainly you had certain things which are more than 95, around 95, and less than that. So what would you really suggest as a cutoff? Again, it's coming mostly from how peaks are identified in by comparing your GC mass spec total ion chromatograms with libraries. But is there a cutoff that you can suggest, or do we need to accumulate much more data to really arrive at such an understanding?
Okay, so well, it's quite difficult questions. For example, if you analyze too much polymer sample, then for example, polystyrene, the peak will be saturated. So depending on your system, let's say a detector, we have to set the calibration range. So let's say also the LOQ as well. So this is depending on your detector too.
Okay. So thank you. I don't see. Okay, so there is a question. Is there any plan to expand the number of microplastic species in your RM and database?
That's a good question, and possibly yes, but I would say it's very difficult to find out a very characteristic peak for one single polymer because sometimes it reacts with other polymers. So we have to consider the secondary reaction among the pyrolyzates. So we need further research to add more polymer compounds in our library.
Okay. So thank you, Ichi-san. I think that was a very nice presentation, and you have actually presented the details of the methodology of PYGCMS for microplastics.